stay connected, we have a link tree up there where you can just scan our QR code and find all of our socials. We will send that out in an email following after this presentation. And to
Hi.
talk about
Oh,
yeah,
oh, sorry. Hi, everyone. My name is Franny. Just before we dive deeper into this meeting, I just wanted to talk a little bit about some etiquette and some norms to make sure this meeting flows nicely. Make sure that your mics are muted unless called on, or if you have a question near the end of this meeting. And it would be great if you guys all had your cameras on to make this meeting more interactive and engaging for everyone in this call. And make sure to prepare some questions because we will have a Q&A session at the end of this Zoom meeting. And thank you for being here. And a little bit about Dr. Brenda Wilson. She has studied many research topics and is a professor of microbiology at the University of Illinois Champaign, sorry. And she's recognized for her research in many different fields, for example, in genomics, host pathogen interactions, and also microbiology, which is very relevant to our collaboration with HUMS, Harvard Undergraduate Microbiology Society. And yeah, and just like a reminder to take notes during the event so that we could have a productive discussion after the presentation. And there's also a possibility that you guys could receive a certificate at the end of this meeting. So you guys can receive a link. So make sure to stay tuned for that. Thank you again. And we'll transition on into into the presentation.
So,
Um,
Mrs. Wilson, you can go ahead now.
Okay, thank you. I was wondering if there was any more that you guys wanted to say first, but thank you all. So let me go ahead and get my screen shared with you. What I was asked is to actually start with a little bit of my background because, of course, I didn't, uh, right after high school, become a professor of microbiology. And actually, I think if you will find out that I don't actually have a degree in microbiology. I have, in fact, never actually taken a course in microbiology before myself. Although I've taught it, I'm a professor of microbiology. It all happened afterwards. And so wanted to share a little bit with you that no matter what your initial path might be, you can change it. You can change it as you go. And when you find your new interests or you actually solidify particular interests that you have, there are always pathways towards a goal that you might have. And so I'm going to tell you just first of all, just start off a little bit of my background, and then I'm going to end with where we are sort of today, because I like to write nowadays actually call my lab sort of an R&D lab, a research and development lab, because we have researched for quite some time now. For a long time we've been studying, and we now are sort of poised at a position where we can actually take what we have learned and actually start implementing it into actual applications for biomedical research and application, actually using it in the clinic. And that is sort of where the goal was to start. And I'm going to just give you a little bit of my background that sort of brought me where I am today. So let me start with that. Okay, so here I am today. Okay, This is the School of Molecular and Cellular Biology, and one of the buildings where I'm in. I share it actually in microbiology plus cell biology is in this building, and then there's a couple of additional buildings behind this one that have other departments in the School of Molecular and Cellular Biology. I am also the director of undergraduate education in our college, and we have about 2,000 majors. That can get degrees in either molecular and cellular biology, biochemistry, specialized curriculum, neuroscience, and now also MCB, molecular and cell biology, plus data science. And so I have a lot of different activities that I have across campus. I also work in the medical college. I'm one of the inaugural professors there. And I also have appointment in the College of Veterinary Medicine, where I serve also as the director of zoonosis research. I am also a senior faculty fellow interfacing with the national labs. So we have quite a few national labs across the country that interface with universities, and these government labs are very important for securing our safety in this country. And then I'm also a fellow of the American Academy of Microbiology, and that is basically the honor society of all professional microbiologists. So where did I start? Well, I actually started as an undergrad at Barnard College, which was a all women's college. It still is today. It's one of the few remaining ones, and it's located in New York City. It also is affiliated with Columbia University. So Barnard College and Columbia College were the two entities. Way back when I was an undergrad, there was no uh, uh, female students at Columbia at that time, but they became co-ed during my time there. But my interests, I had two. So these are the dorms that I stayed in. So I actually lived right here, and I lived right up there, and I lived also over there during my time at Barnard. This is the entry to the main courtyard that you can see here to the left over here. And I had two majors. So I was really interested when I first started. I didn't really know exactly what I wanted to do. I knew I wanted to be some kind of a academic, a professor. That was sort of what I kind of had in mind when I first started. Um, but I didn't know in what. Um, I also have family background in German, so I thought, okay, why don't I do both the liberal arts and the science? And so that was what I did. I started with biochemistry in German, and then I tagged on also a minor in chemistry. Then afterwards, well, actually towards the end of my German, my senior year at Barnard, I actually, one of my German professors came to me and say, hey, I have an opportunity here for you that you can apply for. And that is to actually take into account both your German and your biochemistry, and that is study biochemistry in Germany. And I thought, yay, okay, that sounds great. So I applied to the German Academic Exchange Service, and was a fellow there for a little over a year. And I stayed actually in Munich. And this was the main campus. This is actually the place where I worked was way back in there. But this was sort of what I traveled through daily. It was beautiful. I lived in the Olympia Centrum. So this is where they had the 1972 Olympics, and my apartment was in this building right here. And during my stay there, I actually worked in a virology lab. where we were looking for how you can trace viruses inside people. And at that time, we didn't have fancy things like fluorescent proteins or anything like that. So we actually ended up trying to decorate it with other kind of colorful entities to get it so that it could be used as a tracer during infections. Um, but I also did a lot of exploring in Germany. So even though I didn't have a whole lot of money, I tried to save up my money as much as possible. And I and a friend of mine, the two of us actually literally walked the entire Rhine. So we went by foot all the way from way down near Munich, where it starts, and went all the way up north to where it splits off and goes off to France, and then we went further on to an island in the North Sea, which is where my mother is from. The island is called Fur. But anyway, we enjoyed this. We actually we were able to take pictures of all this and go up along the Rhine, just hiking along there. It was so much fun. And I learned tremendously from that experience. But then, I had to go back. And when I went back, I actually applied to get into uh, uh, grad school, and I did my degree in chemistry. So, and what I was doing is actually sort of at the interface between microbiology and chemistry. So I wasn't totally away from biology. I was very interested in infection biology. I wanted to see how we can actually help with infections and make things better. And I thought, of course, being able to make new antibiotics would be a really good way of doing that. And I was very interested in how they were made and how microbes made them so that we could make them also. And so here I am. That was me way back during my grad school, doing some chemistry, or at least pretending here to do so. Um, and I uh, this is the buildings that I worked in. Every day I walked up these steps here to get to the labs that were over here. This is the other side of the building called Ramson Hall. And Johns Hopkins campus is very beautiful. It's a very nice place, and they actually even spruced it up better, more than they used to when I was there. So it's really nice campus now. And this is the microbe that I worked with, Nocardia, which made an antibiotic called Nocarticin. And this is the structure of it. And this is sort of what I worked on, trying to figure out how it was made and what precursors that you gave to it to make it, to become what it is, what enzymes, the proteins that were used to make it happen, and how could you manipulate them so that you can get various different kinds of nocarticins. And so I worked in Craig Townsend's laboratory. He's very well known in the field of natural product chemistry. And I had my first child during this period of time as well. And it was quite, for me, it was a lot of growth. I learned a lot, not only in terms of the science, but how to navigate the science, how to figure out what my life was going to be like, what I really wanted. And one of the key factors that I realized at the time was that if you're going to be in this field and if you're going to do it, you got to have a passion for it. You cannot do it if you're going to think of it as only as a job or a 9 to 5 job. It has to be your life. You have to really be passionate about what you're doing. And I realized that I put up with all kinds of nonsense to do my work. And it was it really, really made a big difference in terms of the directions that I went, and learned things. So after this, I decided that almost everyone in my lab at the time were all going into industry, and I really wasn't interested that much in industry. Really wanted the academic part of it. I wanted the teaching. I wanted because I actually had to teach a lot during during my graduate work, and I discovered that I really liked it, and I wanted to engage in that, and I wanted to actually help others to also realize their potentials and to see the passion that I had, and that they might also embrace it as well. So that was sort of the path that I ended up taking. Um, I then realized that I actually wanted to go more into the biology aspects of things, looking at therapeutics. I realized that at the time that almost all of my colleagues and my friends that were in the antibiotic area were all going into industry, and very few were actually going into academia, because at the time industry was actually performing all of these kinds of studies and these kinds of discoveries. And so I actually started looking for something else. And what I found was this really, really interesting uh, type of, uh, I guess, virulence factors, but it was something that bacteria made besides antibiotics, but it was toxins. And so I really got interested in the hot toxins, and that's how I ended up actually at Harvard Medical School in the laboratory of John Collier. This is John Collier right here, and this is the whole group that we worked together. Um, and at the time, and this is where I lived right here on in Boston Harbor area, um, and Harbor Point Apartments. That this whole area is, it's near the University of Amherst is over on this side over here. Um, and this is Harvard Square, and we're down in the medical school, and this is the building that I hung out with on the top floor in the building. And it was an absolutely, for someone who loves academia, this was the place that I felt you have to be, because the atmosphere, the intellectual engagement that you had, the thrill of discovering new things. And at the time, one of the new things was actually toxins and how they worked and what they did to animal cells and human cells, and how they did it. And the remarkable thing to me that got me interested in it was that these toxins, which turned out to be proteins, very large proteins, they were soluble, they were secreted or released by the bacteria, and those proteins all on their own could do all the damage to to the host. They so for in the case of diphtheria, for example, so the diphtheria toxin is produced by the Corynebacteria diphtheriae. These are gram-positive organisms. That's why what you see here is a gram stain, very purple color dye that they stain with. And they get infected with a phage, which is a bacteriophage, that's like a virus that infects bacteria. And as a result of that, on that phage is encoded the gene for a toxin. And it's a single protein, but it has multiple functions. And that was one of the things that struck me with these, is that they all alone can bind to receptors on the surface of host cells, and by host cells I mean human or animal cells, and then they can get taken up into them. And when they get in, they then can also get out of the vesicles that they get in and enter into the cytosol, where they react with their target molecule and cause all kinds of damage. And in the case of diphtheria toxin, that was basically killing a cell. And one molecule, one protein, which is an enzyme, gets into a host cell and it will kill it. Only one is needed. It's very potent. And I just thought, oh, that's really interesting. And then the target that it has is actually something that is unique to eukaryotes. So it's a modified amino acid, and it reacts with another compound. So there's two substrates that this enzyme catalyzes the reaction with. And I just thought that that was the most fascinating thing ever. I loved it. I did a lot of work with that. And I decided that this is sort of the direction that I wanted to go. And when I went to my first faculty position, I decided to continue in the direction of toxins. But I changed the toxin that I was working on. I actually started working on a different one. So my first faculty position, where I actually then also was promoted and got tenure as an associate professor during that period of time, was at Wright State University, um, basically named after Orville and Wilbur Wright, the Wright brothers. And it's located in Dayton, Ohio. Um, and I started working on a brand new toxin that was made by an organism called Pasteurella meltasta. And my interest actually from it is that I have a vendetta to fill because this organism almost killed me. So I actually ended up in Boston. Those of you who might be from Boston know of Mass General Hospital. So right next to it is what they call the Ioneer, Mass General Ioneer, which is a hospital for those that have ear, nose, and throat problems. Well, I ended up being with a really bad infection that started in my throat and tonsils with Pasteurella meltasta toxin. And as a result of that, I actually lost partial hearing. So I don't have complete hearing anymore because of this organism. And then I found out that not only could I also have, gotten deaf from it, but oh my goodness, I could have also gotten cancer because it turns out that this toxin actually causes cancer-like effects on cells. And it does that in all kinds of different animals. So domestic animals, pigs, get a disease where the nasal and these are the turbinate bones inside the snout of the animals, basically atrophy, they go away. And it's caused by the toxin, not the microorganism, but actually the toxin that is made by it. And then we started studying it. And one of the things that we wanted to know is that, well, what is its target? How does it work? And it works by just like what I showed before with diphtheria toxin, or I saw that it gets into the cells. And once it gets into the cell, it actually causes all kinds of single transduction changes in the cells, which in some cases could change the cells and their morphology. You can see here, these are some cells. You can see that if they were labeled with a green fluorescent protein, they can get into the cells. The red that you see is actin inside cells, and you can see that they get all, you know, they change their shape, even though this is a normal cell. But look what happens in cells that the toxins is in. So lots of different changes are happening with them. And if you give it to, say, cells that could differentiate into fat cells, and the fat that you see here is the red, is dyed with red. This is the normal pre-adipocyte, which is a pre-fat cell. Um, and you differentiate it into a mature fat cell. It gets lots of fat droplets in it. Treat it with toxin, however, even without any differentiation, you get all kinds of morphology changes, very much like a tumor generating lots and lots of cells. If you have differentiation media that would normally cause it to differentiate into a fat cell, if you treat it with that, it turns out that it actually still not a normal cell, but it actually can help reverse the differentiation. So it prevents it from becoming a fat cell, but it still causes it to grow. And these were some of the things that we learned during that process. And that was part of the intriguing aspects of this for me, because I thought it was just really fascinating that one protein can do all of this stuff. And the protein is made by a bacterium that, what is the point of the bacteria making it, right? Everybody always asks, why does the bacteria make these toxins? Well, we think, all right, we now have a lot of evidence that it actually has some effects on the immune system's response to the bacteria, and that's really why it helps the bacteria actually survive inside host cells. But we didn't know that at that time when we first started, and of course, I wanted to grow even further. I wanted to do even more. Um, and after I got my tenure at Wright State, I actually got recruited to the University of Illinois. Um, and I've been there ever since. So this was in 1999, and I've been here now ever since, doing all kinds of amazing things. The growth that I had and the interest that I was able to have and the resources just really allowed my program to grow. Um, I had a lot of very excellent students that came along and helped with this process. And so not only were we working on toxins and finding out what aspects the toxins had, we were learning from their evolution. We were learning from the genomes that they had, looking at various interactions of proteins inside them, and understanding how infection occurs and the role that these toxins play during that infection, and then just doing lots of genome comparisons to find other uh, bacteria that make these toxins, and being able to understand them in the context of evolution, but also we started understanding a lot of what was happening with them so that we could actually start thinking about making and converting these okay, these not friends of ours, into friends by using them for good purposes and getting them to actually do some of the work that we would like to do. During this process, I also want to say, and I'm not going to talk very much about this, but my lab expanded quite a bit when I came to Illinois. And I also was one of the initial theme leaders of the host microbe systems theme. We were actually working on microbiomes before they were called that. Um, and we still are. So we still are working on looking at human and non-human primate coevolution with their microbes, and we're studying also microbiomes and how microbiomes in the environment impact us through food and through other mechanisms and our health. And so, but I'm not going to talk about that, but that is one other aspect that we work on. But instead, I'm going to talk a little bit about what we're doing in terms of how we're taking the information that we got from toxins and now starting to use them to deliver other things, to get them to deliver therapeutic cargo that will actually help in hopefully curing some diseases. So that's what I'm going to start with. So that's what led to the idea. There's toxins everywhere. We have there's so many genomes out there now of microbes that have toxin modules in them. That is pieces of genes that actually code for um, some of the functional aspects of these toxins, and that's where we started to think about, well, how do we turn a toxin foe into a friend? So what I'm going to do right now is just give you a little overview of, well, how do we do it? How do protein toxins that we work on act on cells? How do they do that? And then how can we use them right now? What are some of the applications that toxins are being used for? And I'm sure that most of you have heard of Botox. It's a natural product that is made by Clostridia bacteria, and it is now being very widely used for both cosmetic as well as therapeutic applications. And in our studies, we now are looking at developing platforms that we have named bacterial toxin inspired drug delivery. These are our first prototypes were done for post-exposure therapeutics against botulism. That is the paralysis that you get from Botox. Um, sorry, and then harnessing some of the evolution to actually help, um, sorry, and then harnessing some of the evolution to actually help better design these so that we can deliver the therapeutics. So to start, toxins are modular proteins. So what I mean by that is they have parts to them, different parts that actually have different functions. They do different things. So in most toxins that are made, they have two parts. One part is the cargo, that is the toxic part, the part that actually cause some kind of damage to a host when it gets in. And then there's the delivery vehicle. And that usually can have multiple subparts to it, but the function of it is to actually deliver the cargo into the host cell. And in this process, usually it recognizes a very specific receptor on the host cell, and its interaction with that host cell results in this cargo being delivered into this inside of the cell, where it then interacts with a target, and then it does its thing to cause some kind of changes in that cell. And one of the things that we have discovered and explored for our studies is that these toxins are very modular and swappable. So you can take, an A part from one toxin and you can put it onto another toxin's B part, and you now get a different functionality. And when that happens, basically what you're enabling is a possibility of having all kinds of building blocks, cassettes that you can swap with. Just like, with Legos, you can swap all the different pieces together and make different things. Well, we can now do that in real life with these toxin modules, and we can do it using biotechnology, gene swapping, and we can actually create what we need to uh, elicit the types of things that we want. And importantly, we can change this cargo. We can make it so that the cargo is no longer toxic, and instead, we can swap it out for something else that we want to deliver inside the cell, say like a drug of some sort. So, um, So in each of these cases, there's usually two types of these kinds of toxins. The ones that we in my lab mostly work on are the ones that are shown here on the left, but there are other toxins that also can work, and they come together. They're usually separate pieces that come together as a complex. A cholera toxin, anthrax toxin, pertussis toxins. Those are, I hope, somewhat familiar to you all. But we focus mostly on these toxins over here, and in particular, the botulinum neurotoxins. All of these get in, and they're a single polypeptide chain. They're made as a single piece that has multiple domains, and that's basically what the is. And when they get in, they can get out at different places, and they can interact with their whatever target inside the cell and do some cellular effect in there. And so what we have uh, noticed is that you can use these toxins for a lot of different applications. Because they're modular, they have the toxic part, which a lot of people have dubbed as the warhead. Um, they have a delivery vehicle part, which is basically a very specific binder of a target cell. And that target cell can be any receptor that is uh, uh, on a particular cell that you want to go to. Say, for example, a cancer cell, you can target it very specifically by changing this part of it so it recognizes it. And all of these can have different activities. So the activity domain can be a cell killing activity, or it can be a cell changing activity. And it can recognize different receptors through, hormone receptor It recognizes the same thing, same receptor as a hormone. It recognizes antibodies can bind very specifically to a target receptor. And all of these can be swapped around and utilized for applications. So, all right, so what are some of the applications? Well, one of them is actually taking advantage of the cell killing toxins. And we have now a number of immunotoxins. In fact, we're actually in the hundreds now. When I first made this slide, it was only about 100, but now we've got about 300 or 400 of these clinical trials that are ongoing currently to actually have ways of getting and delivering these warheads. These are the cell killing domains of toxins, getting them inside a cancer cell where they muck up the cancer cell and cause it to die. And that basically allows for killing off tumors. And we now have quite a few of these that are on the market um, for different types of cancers. And there's many more that are being developed and are in the clinical trial areas. But different ways of doing it, different types of approaches. The idea is you have the cargo that is going to kill the cell, you have the delivery vehicle, which allows it to get inside and get to where it needs to go to kill it. All right, so when we first started working on this, we actually felt that we wanted to actually use something that we could actually have a system that we could manipulate and use and work with that we could actually have good controls for, so that we knew we were on the right path if we got it right. So we started with the botulinum neurotoxins, and these toxins are not cell-killing toxins. So they actually just change the cells. And importantly, if you are able to survive the toxin treatment by, basically being on a ventilator and getting intravenous, food and stuff, you can actually last long enough, survive. It will revert back, and so you can regenerate. And in that process, you can actually then know what has happened, and you can monitor it and follow it. But botulinum neurotoxins have good parts and bad parts to them. They're what we call true dual use biological agents, and they're responsible for food poisoning, botulism, the paralysis, and they have also even been used as bioweapons. Okay. And I'm going to tell you a little bit about some of that, but I also am very excited about the good sides of these. Now, of course, they've been used as cosmetics to, get rid of wrinkles, and I'm sure everyone's heard of Botox and being able to do that. But there's all kinds of other therapeutic agents, activities that you can use them for, and I'm going to show you a couple of the what I think are really exciting applications. So first of all, paralysis. Okay, the paralysis is the bad side of this. They're incredibly potent. Very, very little is needed. We're talking picograms here to kill a mouse. So very tiny, minute amounts of it to actually cause damage. And they're made by Clostridia, which are spore-forming bacteria, so they can last for a long time. And usually the way most folks get exposed to it is through eating something that is contaminated with the spores. And here in the States, most of the time it is seen as because organic foods that are not pasteurized well enough haven't been treated to get rid of the toxin. Usually, you can result in what we call floppy baby syndrome, or just paralysis, as you see in this 13-year-old girl here. Or it can be by accidental treatment with Botox to get rid of wrinkles, and you overdose. You can do that too. So the toxin itself is a large protein. It's 150 kilodaltons, so it's got 1,295 amino acids, and each part of it is 50 kilodaltons each. So this is the catalytic part. this is the part that gets the catalytic part across the membrane of the neuron cell, and this is the receptor binding part. Okay, so these two regions here, this is called the delivery vehicle, and this is the cargo. And the cargo is held on to the delivery vehicle using a belt that is shown here in green. You can see it here. So here's a space filling model so that you can see how it's shaped. And here is the green part. The green part that's in the back is what you see right here. And then it's going to turn around, and this is the active site right here that you can see. And you can see that it basically here it inserts into the active site right there, and it wraps around and basically holds it close, and only releases it once it gets into the cell that it's targeted. So how does it work? Well, in a normal neurons, right, when you have normal action, you have the neuron coming on top of the muscle, and here's the muscle cell, and the muscle cell has some receptors on it that need to get the neurotransmitter. And the neurotransmitter is usually held in vesicles that, when they get an action potential, a signal that sends it to go to get moving, it then has these vesicles that then fuse with the membrane and release the neurotransmitter, and then it goes to the muscle cell, and it causes you to twitch or whatever in your muscles. Well, what happens in botulinum is that the toxin gets in to this presynaptic nerve terminus. It gets in and it basically chops things up. So it cuts proteins that are important for this fusion event to occur. It doesn't happen. There's no neurotransmitter release. All of these vesicles that have neurotransmitters sort of pile up at the end, and you can't get anything moving, and so you are paralyzed as a result of that. And a good application of this is basically that you can, with Botox, you get, release of a lot of the wrinkles that you might have. Well, if you use it for wound healing, which I think is a really exciting application that is now available, is that you can actually now make it so that there's less scarring. And here's an example where you have a very bad lash to the eyebrow, and after a year, you can see that you can barely see the line here because there's hardly any scarring that happened. In the control person, the lash the scarring that had happened here, there's lots of scarring that resulted, and that is because your muscles are so tight when you are recovering that they scrunch up and they actually cause scarring to happen. Whereas if you have Botox treatment, you can actually relax your muscles, and the scarring will mend without having all of that happening. So I'm very excited about that. There's now quite a few papers on this that you can look it up. It's actually gaining a little bit of momentum. And same thing for things like keloids. So keloids, there's collagen, excessive collagen buildup because of where there's might be a scar. This probably resulted in a pulled earring or something that happened here. And 1 year later, after treatment with Botox and sometimes some steroids, you can actually get it to where there's no more keloids. Okay, so it's very exciting, positive applications, therapeutic applications of toxins. All right, so what we decided is to use this system, and now instead of delivering toxic part, which will cause paralysis or whatever, let's see if we can actually deliver other kinds of cargo. And of course, why are we doing this? It's because the biggest challenge right now in the pharmaceutical industry is that there are all kinds of antibodies and therapeutic enzymes and other kinds of protein drugs that are out there, but they can't get into the cytosol that can do it. We have many toxins that can do that. Um, and what about how about we swap that toxic part with a drug for applications? So we decided to use the botulinum neurotoxin system as a test thing here, and looking at botulinum neurotoxin inhibitors. And why was that actually, why is that something that's important? Well, it turns out that there are no therapeutics. You have botulism, you're stuck. You can't do anything but slowly self recover. You have to be on ventilators, you have to get intravenous feeding because you're paralyzed. Once the toxin gets into the cell, you are paralyzed. And right now, the only thing that is used for toxin mediated diseases is what we call passive immunization, which is basically you give some antibodies that, in case of most humans, it's horse antibodies um, that will prevent the toxin from binding to the cells and further paralysis from happening. There is some humanized antibodies available. We call these baby big, and they are very expensive. And there's only one place you can get them, and that is the California Department of Health. And you have to request permission from the CDC to get them. And there's a lot of delays in getting them to the help, but right now it is restricted to floppy baby syndrome for use. So we wanted to fix that problem. We targeted that as our goal, and the idea here is, can we get a delivery vehicle that will deliver the therapeutic into the cell um, where the action is needed? Okay. And of course, the idea was inhibitors of some sort, antibodies that would block the toxin action. And of course, our prototype that we would use for this would be a green fluorescent protein that we could actually see. And that's what we did. So the idea here is, you get your inhibitor up here, you have your catalytic cargo and everything. And there has been applications, there have been some in the last, I don't know, 3 or 4 years, there have been some studies where they actually just piggybacked the inhibitor onto the full length toxin by just cutting out the pieces in the middle here, which are the part that actually causes the toxicity. By cutting that out, they were able to do that. But of course, the problem is you put the inhibitor up here and you've still got huge amount of protein there, which makes it more cumbersome and more difficult to purify and to get it actually applied to people. So we wanted to just do this. We wanted to have any cargo, and we started with just GFP so that we could figure out how to connect it to the delivery vehicle and try to get it to work. And we were able to do that. So we actually had, with and without the translocation part of the domain, we could deliver GFP into the cytosol, no problem. We could do it in tissue culture into neuronal cells. So you can see here that the green is inside the neurons cells. And we could do it in vivo. So we could do it in a mouse. All right. So after 3 days injection into a mouse, we actually were able to show that indeed we could get the toxin delivered to the nerve termini. So here's what I'm talking about. So the botulinum neurotoxin is up here, okay, on the presynapse, and the postsynapse is down here. And what we're seeing here is we have a red label that actually recognizes the receptors on the bottom here. Okay. And what you're seeing is a layered sandwich here because the green is actually on the presynapse. So you have two layers. But this is just the receptor binding part. This is not the part that actually gets it across the membrane layer. So it's not the part that allows it to enter into the cytosol. We could actually, if we include the translocation domain, we can actually get it. You can see here that it's now diffuse throughout the cell, because it not only got into the vesicles, it got into here, but it actually got out in here as well. So we were very excited about that, and we started saying, okay, we have uh, a delivery vehicle. Now let's swap in the inhibitor. And we did both a substrate, based inhibitor, as well as an antibody. Okay, so an antibody, the binding part of an antibody, we hooked it up to this, and we tested it out. So of course, the big deal here is, well, can you actually prevent paralysis? Can you reverse the paralysis part? Well, so what we did is we did that again in mice. And what you can see here is we used two different assays for this. One is called the toe spread reflex assay. This basically uh, is that you inject the toxin into one of the hind legs, and you look to see which one it inhibits, and you use a test. So it's like a reflex, just like when you bonk your knee, when the doctor bonks your knee and your knee flies up. Well, in mice, if you raise their tails, they can't do anything but have their paws go like that. And so that's what you see here. The tail is being raised, and you can see that the one where there is no Botox spreads out, but the one where you have injected Botox, it doesn't spread out. And notice how little was added. Attomoles, not molar, moles of Botox was injected here. Okay. The other performance assay is keeping the mouse on a rotor wheel. So the rotor wheel slowly moves, and the mouse has to stay on. And there's a bed of straw on the bottom here, so it's not going to hurt them. But you can see that it's paralyzed, so it can't move. It can only use the leg that actually is still working. It is not paralyzed. And you wait to see how long they stay on the rod. Well, here's what one of our first studies, where we basically injected with Botox to paralyze. 2 days later, they're completely paralyzed. On the 5th day, we injected with antidote. All right. So basically, what we're saying is we want to recover from paralysis. And what we're seeing here is, with or without that translocation domain, the one that gets it into the cell, um, you can see here that with it is quicker, it recovers faster than if you don't have that translocation domain, okay? Which was great. And you can see here over time, the results that we're saying here. So with the full antitoxin, day 1, there's no recovery. This is paralyzed, and here's the one that is control. After 4 days, you don't really see a whole lot happening, okay? And you certainly don't see anything happening in the other two controls. But by day 8, the one that is the actual antitoxin, you start seeing recovery. So all of these are the same mouse that's being looked at. And you do the performance assay, and lo and behold, you get recovery. So the one that has the full antitoxin, any of the other controls are not recovering nearly as quickly. So we're now at the point where we're thinking, okay, let's put on any other cargo. Can we do other things with it? And we're certainly moving forward with that. We are thinking not only in terms of warheads that can target cells for destruction or changing them, but we're now looking to see, are there other things that we can do, that we can actually modify things? So can we modify with enzymes or effectors? Can we use gene editing tools? One of the things that we've been able to show is we can have a gene editing tool that actually will get HIV cut out of HIV positive cells. So Can we deliver those? And those are part of the directions that we're going for, looking for target specific medicines, optimizing these delivery vehicles, finding targets and being able to link them together so that they can be stable proteins that can be made in large quantities, purified and used for humans even. And of course, I didn't do all of this. I have a huge number of students and folks that have worked on these projects with me. It's always been a lot of fun. I had some collaborators that helped with a lot of the neurobiology part. And of course, I wouldn't be anywhere without funding from our national labs and agencies. And so I will end there. For any questions that you might have. And I don't see, I'm going to stop sharing so that I can see if there's anything in the chat box for you guys. Well, I think so. In terms of what's the most exciting or emerging areas of microbiology, I think that being able to use microbes for therapeutic applications is actually very, very exciting. So microbiomes, for example, by modulating your microbiome, by actually treating it or even introducing a healthy microbiome into situation, being able to actually use that to help with a lot of different kinds of chronic diseases. I think that that's a very exciting area. I think, well, of course, I'm very excited about the application of using toxins for therapeutic deliveries. Um, but I also think that gene therapy tools, so gene editing, are also a very exciting area of application. And I was thrilled when we finally saw that RNA vaccines were applicable. So I saw the struggle in the early days, in the early 2000s, when people started trying to get RNA vaccines to be able to use them. And now we actually are using them, and it's been a long haul to do that. So I think that there's so many directions that microbiology can go in terms of how it can help us, but also in what we can do with it. So I don't think a lot of people realize that there are so many applications in the food industry, in the textile industry, in the materials industry, where microbiology is very, very important. Energy, microbes. When COVID hit the University of Illinois, we actually had a really major problem with being able to use alcohol to for disinfectant. All right, so our fermentation, we had in crop sciences, we had fermentation for bioenergy fuel development. They converted all of that into actually making alcohol so that we could use it in our labs and for other purposes, and even to give it for first responders and stuff. So it was amazing how the things that you don't realize that are very dependent on microbiology, energy, all of our, the carbon cycle, everything that we breathe is very dependent on microbiology, and having folks that are really knowledgeable and can be innovative in using those, I think is very important. Right? How does Botox essentially get rid of the keloids? I think that the big way that it does that is it actually acts on the nerves that are there. So one of the problems of generating the keloids is it's kind of like the scar tissue that show that it tenses up, but also what happens is uh, the immune cells that actually stimulate the keloids from happening, it causes a lot of inflammation. Our system can carry. We have it can carry CRISPR. The challenge right now, and we're working on that, is it being able to carry the guide RNA as well. So we that part needs to still be worked out in terms of being able to use toxins to deliver gene editing tools. So if you can get both of those happening, and there is some ways now that are the technology now is moving in that direction. So I'm hopeful that will be the case. All right. What is your biggest advice for students who are just getting interested in microbiology? Explore. My biggest advice is explore it. Find out what is cool about it. What are the things that microbiology is available to there? I mean, not everyone is excited about the same things. That's okay. It's good to be excited about different things, but see which areas are exciting to you, and see how that actually might benefit. Um, I mean, like I said, I never took a microbiology course in my life, but I find it later. I realized that, oh my gosh, it is so important. It's so cool. And I then caught up with all of that. I started learning and learning a lot about it. And my interests did change with time. I mean, I learned a lot about some things, and then I grabbed hold of something else, and all of a sudden I was excited about that too. Um, so I think that is um, definitely a way to check it out to see if that's something that you really are interested in. How is biological research different today from when I was a student? Genomes, sequencing. I didn't have anything like that. So the way I sequenced genes was by the old Maxim Gilbert method. So if you don't know what that is, look it up, because you will be astonished at how long it took to get any sequencing information from doing Max and Gilbert. So first of all, you had to be a chemist because you were doing chemistry, real actual chemistry. And once you did the reactions, you also then had to run these ginormous gels, which were 5 foot tall. And you had to put the little each of the reaction mixes in the top of the gel, and you had to run them basically overnight. Um, and then you had to, oh, so very carefully remove them from that glass plate. And if your glass plate was dirty, you were messed up, because you had to do it all over again, because it wouldn't work. And then you had to put it on X-ray film. And of course, you were all the time using radioactive material. So you had to get all, gowned up, to work with that, right? And then you had to wait 2 months to get your results. So it was very frustrating and long and arduous. Now it's so different. And I can tell folks right now that most of the techniques that you do that you're learning now, 10 years from now, they're probably going to be obsolete. So if you're exploring labs and doing different things just to learn the techniques, that is not the way to go, because nothing that I did back then are we doing now. Okay. um, and I told you a little bit about the virus that we were working on. We didn't have green fluorescent protein when I did that, but now we have green fluorescent protein. It's so many things that are different. And I think that the biggest thing is to learn the fundamentals, learn the critical thinking and the problem solving. And I can guarantee if you focus on that, you're going to be very successful in almost everything that you do. Would you recommend people who are trying to get into microbiology, specifically your research topic, to start with? It could be seen intimidating in the beginning. Yes, it can be, but you know what? I was very intimidated when I first started too. I didn't know what I wanted to do, but it turned out that at every step of every tier that I went to, yeah, the very first year of it was so intimidating. You feel like an imposter. It's like, well, how did I end up here? Oh my gosh, this is overwhelming. It's terrible. And and then you get used to it. You learn more and you accommodate and then it turns out that by the end, when you're ready to finish, and so it's okay. And everyone notice. I have a lot of students in my lab who's just starting off, and you can tell that they're scared and they're self-conscious, but the more they do it, the more they practice, the growth is just phenomenal, and you can get so much out of it. And I think everybody, um, when it's time to get to the next step, you're going to be ready. You just need to plow through it, stay steady, and the key here is to engage, always to engage intellectually and with commitment of what you're doing. And I think that you will persevere. So I think starting with the fundamentals, and the thing is, and the reason why I say fundamentals is because if you have all the fundamentals, you can switch gears at any time afterwards. I mean, uh, my PhD's in chemistry, right? My first faculty job was biochemistry. My second faculty job was microbiology. And I had actually, when I was interviewing here, one of the interviewers asked me, so you've never really done very much microbiology or anything. Why are you applying to a microbiology department? I said, because that's where my heart is. That's where my passion led me. And well, I got my job, right? So, but really, that's the key: is that if you have the foundations of what you need, then you can switch gears. One of my very dear friends, who was also faculty in my department, her name was Abigail Salyers. She ended up being president of the American Society for Microbiology. Um, she was a professor of physics, yeah, and said, "I don't like this anymore." She was actually pretty well known. She had nice publications and everything in physical, in physics. And she said, "I don't want to do this anymore." And she said, "Okay, so what did she do?" Well, she switched gears. She went to a lab that would take her as a sort of a senior postdoc type of scientist, and she got some training in microbiology, and then she got her job here, and she became president of ASM. She herself never actually took a microbiology course, right? And but so what that means is that if you have the tools to know and follow what your passion is, and the tools to figure out and learn, you're going to be fine. And really, it's setting goals and trying to meet them. And one of the things that I always like to say to folks: start at the top and work your way down, not the other way around. Try it. Maybe you won't reach the top, but you might reach the second or third rung, you know. And if it doesn't work out, well, okay, go to the next one. But the thing is, you got to try, and then when you get it, go wholehearted into it, and see if this is really truly what you want, and it will show. It's very obvious to me when someone, a student comes into my lab, and I can tell when they love what they're doing. It is obvious that they like it. They're committed, they're engaged, they're learning, they're asking questions. That's a key. And I think that you're going to be very successful if that happens. What is the advantage of immunotoxins in comparison to other cancer treatments? Well, the other cancer treatments are radiation and surgery. So I would say that right now we don't have immunotoxins available for all cancers. They're getting there. They're ranking up in terms of which ones they can treat. But radiation therapy is horrible. It's extremely painful. Immunotoxin therapy is not, okay? It is not that bad. Yes, you do have reactions. Yes, you are not well, okay? But it's not quite the same thing. And you can have bad reactions to it. Um, and so so it's just a matter of, what has been tested, what type of cancer you have, what the target is, and seeing what can be happening. Now, some of the CAR T inhibitors and things that they have now, those are actually kind of has the same idea, but it's not quite the same in that those are actually modifying the immune system so that you can use other mechanisms to kill off the cancer cells. So I think that immunotoxins have a lot of advantages. The problem is we don't have a big enough library of them yet. Um, so they're against the ones that are most common and the ones that are easiest to direct at. And I hope that more and more will come on the um, uh, be able to do it. Now, in terms of gene swapping, well, basically gene swapping is you take a sequence and you digest, take a piece of it and you basically clone it into another piece. So there's a lot. Basically it's recombinant. It's biotechnology. And molecular biology types of tools. You can look at that sort of thing up for cloning. It's gene cloning, but now what you're doing is using pieces of a gene rather than the whole gene to swap. Could AB toxins be used in the future as alternative to antibiotics? Yes and no. So it depends on what the application is again. So the use of these toxins, so for example, the ones that might be targeting a type of activity for delivery of something, that could work if what if you can't use an antibiotic, or if the antibiotics or that you're or the microbe that you're trying to to mitigate the harm from, um, if you are, say for example, cholera toxin. So cholera is caused by a bacterium, but you can actually now get something that will actually inhibit it, then you can use it. But that would not be the same thing as using an antibiotic. Antibiotics are not quite the same thing. And I would say that these are um, two different types of applications, uh, killing the microbe versus killing the host cell. Not the same. Counteracting the action of a microbe on a host cell. That's what you could use a immunotoxin or an AB toxin to do. It's not the same thing as an antibiotic. Yes. So could a beat it be used for editing or priming? Yes, you could, because the idea is you have a whatever your cargo is, that's the biologic that you want delivered. The thing that the beat it does is it delivers that cargo. So if you can get what you're looking for into the form of a cargo, you can definitely be able to deliver it. And I'm pretty sure that we could make it so that most things will be able to be delivered. We're working on many aspects of that right now. So the bigger the repertoire of tools that you have, the I think the greater the potential. What was my biggest takeaway from my time studying biochemistry in Germany? Oh, well, one. So so it was actually for me. It was very interesting. So when I was learning chemistry, one of the things that I learned was that there was a form of a cargo you can definitely be able to deliver it, and I'm pretty sure that we could make it so that most things will get be able to be delivered. We're working on many aspects of that right now. So the bigger the repertoire of tools that you have, the I think the greater the potential. What was my biggest takeaway from my time studying biochemistry in Germany? Oh, well, one. So so it was actually for me. It was very interesting. So when I was learning chemistry, one of the things that I learned was that there was basically it's like an encyclopedia of organic chemistry. It was a book that everybody went to. It's like the holy grail of you know uh, of organic reactions, and it was called Beilstein. So this was where they just compiled all the different things that you could do with Beilstein. So in going to Germany, I had envisioned that oh my gosh, I'm going to the seat of where all this stuff had happened, the history. It was amazing to see the history of where that all originated. So while I was there, I went to a lot of museums. I went to a lot of art exhibits and lots of music exhibits and the culture. And I could see, and one of the other things that I could see, it was on a very personal level, was I now finally understood my mother. My mom was German. And for when I was growing up, there were a lot of things I didn't quite understand about her mentality, the things that she did and the way she thought. And after going there, I learned that. And my time when I was there learning and studying, um, all of the personal things that I also encountered while I was there really made that experience incredibly meaningful. And it also solidified my interest. So I loved being able to figure out how to decorate viruses with something that you could then trace in a body. That whole idea. And then I realized that, oh my gosh, it's so much tedious work that you have to do it. I realized that the idea and actually the work that you needed to accomplish that idea, and this was the first time I realized that that was something that had to be done. And it really helped me a great deal in my graduate work because I realized that all of those tedious things that I was doing that just seemed like it was endless. I got to go into the lab and do again, repeat all of that. I had to do more, more colony picking, more, measuring things, more waiting on something to develop, which is very, boring. If you're just sitting there waiting for something to happen, or you're just doing something very menial, and you're thinking, why am I doing this? And then for the outcome that you have, and you realize you have to set the goal for the outcome and not what you're doing along the way. And so later on, when all I was doing was sitting in front of an HPLC and injecting things to look at what happened in the as it was going, and every time and monitoring it, make sure it didn't break down, or every time that I would pick colonies and umpteen thousand colonies, petri dishes and plates, all those times, those were tasks that were so you could think of as menial and time consuming and tedious. But in the end, you realize the result and you're excited about it. And it was just that realization that you got to do the work to get the results. That was, for me, that was the enlightening thing. Uh, do I think that we will stay ahead? Yes. Why? Because if we have people that like you guys that are interested in this sort of thing, that you are so passionate about this topic and that you want to do this and you are focused on doing the science and you stay focused on the science, we're going to be able to be successful. um, and that's the key. There are an awful lot of my colleagues and a lot of people that are working here. All of us are plowing ahead, staying on track with what our science is telling us and what we're doing. We're doing it for science. We're not doing it for any other reason. You got to stay focused on that. You got to realize that the end goal is going to be something worthwhile. And if it is, if you can see that the end goal is going to be something that we want to do, and you stay focused on that path, it will be so worth it. I can't say anything more than that. Um, I would hope that that would continue. I hope that the funding will continue so that it's possible. Um, but you know, we went through other periods of time when the funding was poor, when the funding for science was down. I lived through some of that where the chances of getting a grant was like 1 in 20 or 1 in 30. And it was hard constantly having to apply, begging for money to do the work that you wanted to do. You got very inventive in terms of making do with less than ideal conditions. But you know what? You still stay focused on the science, and in the end, you're still ahead. So you have to stay positive. Being positive is the key here. The biggest challenge that we have right now with actually our design mechanism and things that we're doing is optimizing it. So there's several. There's we proof of concept is there. We have the proof of concept. But in any time that you develop or you do something, that proof of concept um, needs to be optimized. So that's where the challenge is, because you got to make it so that you can make a lot of the protein, you can make a lot of it pure, that it's stable, that it can be formulated into something that you can use in the clinic, that it actually most of it gets into the cell efficiently, so you can don't have to use gobs of it. You need to use small amounts of it. You have to optimize the delivery mechanism. And right now, optimization of delivery mechanism is one of the things that we're actually focusing a lot on. So being able to hook up the cargo to the delivery vehicle in a way that it will actually get delivered into the cytosol without going off and getting degraded, or not being efficient in getting out of the endosome where it initially goes in. Um, and we've actually made some strides in that regard, actually figuring out some of that. So I think that that's where right now that we're focusing on is optimizing it so that we can actually use it. And I think that that's it's moving forward, but it's still work that needs to be done. Yes, using toxins for as anti-cancers is the way to go, and there's actually quite a bit of applications now for it, where you could actually deliver uh, silencing RNA, so RNA that inhibits the production of cancer genes, and so that you don't get the cancer proteins being made. And those are definitely there, and there has been success in that arena. Well, the so baby big, the cost of baby big is because those are real human antibodies that you have to harvest from people who have gotten Botox. So um, that whole process of collecting serum from people, so that you can collect the antibodies from those individuals and most of them are volunteers, or you have to pay them to have them give you their blood. That is the cost that you have there. So, is it possible? Maybe. I know that there are some people who do it voluntarily because they believe in it. So they, if they have lots of antibodies, the one of the ways that we've been trying to do that is actually genetically engineer so that you can make the humanized version of the antibody. And I think that that is one way to go. And I think that that's probably the direction that some folks are moving in, to make it so that you can make a truly humanized version of it. So the key about humanized antibodies is that they get post-translationally modified. So they get modified after they're actually made. And they get modified specifically and specially in human cells. And so there's all kinds of regulation that goes on in that. Can you use human, cells to make things that you're going to use in somebody else? There's a lot of stuff that has to be worked out there, and I think that people are trying, um, but there has to be. It's not fast. It's going to be slow go. I think I answered. Anybody that I missed?
Yeah, I think that's all for Q&As. Thank you so much.
Great.
Okay, we'll just end this really quick. I'm just gonna go through some of the slides. So, last, we just wanted to say how to earn a certificate. As mentioned before, we will send out a quick quiz in the following week. And if you earn a passing score of 70%, you'll receive a certificate signed by the professors, STEM2Connect, and HUMS. And you'll also receive the certificate through an email after you complete the quiz. And so, yeah, thank you guys for all coming today. And a huge thank you to Dr. Wilson for offering her time and being so generous with the information. Yeah, and so if you're, please promote our event among people who are interested in STEM subjects so we can kind of spread our initiatives and stay active. And we'll also inform you about future events on the Discord channel and on our social media. So yeah, follow our social medias. And there's also going to be a feedback form sent out. As you guys probably know, this is our first ever event. So we're still working on a lot of things. If you have any ideas or any feedbacks that could potentially improve our organization, we would appreciate and love that. Yeah, thank you guys again. And that's it for today.